Everything great? One student asking, discuss paper, sir. Paper is there? Gift paper is there? Yes. In board?
Pieces. Piece in piece.
Open it. Yeah, I'm fine. Thank you. Gold not, gift paper. Gift paper is not there, na? So paper is not there in the board. So you can ask that question, I will say. Paper was easy, difficult, moderate. How was the paper? First tell me. Lateral movement in cell membrane is due to, okay, I'm going to tell it. Any other question? Blue-green algae is prokaryote or eukaryote? Okay, see the things. So, I am telling you about the lateral movement. See, basically, NCERT wrote a line that lateral movement is due to fluid nature of the lateral movement. NCERT wrote, if you have the textbook, it is due to fluid nature of lipid present in cell membrane. Got the idea? I told you about कि plasma membrane have PM, in short I am writing, have a quasi-fluid structure. And in this quasi-fluid structure, who is behind this quasi-fluid structure? Lipid is behind the quasi-fluid structure. So in this lipid, suppose that it is lipid part of the plasma membrane. Assume it is fluid part of the plasma membrane. And in this fluid part, who is moving? This protein is moving. Protein is showing lateral movement. Actually, what happens? Something attached with the protein here. Then protein going for 180 degree rotation. It is the receptor. It rotates by 180 degree and this side came this side. Got the idea? So rotation possible what? When this part going to, anything is rotating. First face is this side. Then it is becoming this side. Then it is becoming this side. So it is possible due to which particular type of movement? Lateral movement. So lateral movement of protein, lateral movement of the protein. Who going for lateral movement? Protein going for lateral movement occur in which one? Lipid bilayer. Lipid bilayer. So fluid nature provided by lipid, and who is moving? You recall a class I was saying you, if I take a nail and hammer and in a solid I just going to penetrate it, can I drag it? No. But in honey, if I take a stick, can I rotate? Yes. So honey-like structure formed by which one? Lipid. Got the idea? Honey-like structure formed by which one? Lipid. So this protein, this is protein. This protein is showing lateral movement in lipid bilayer. This is lipid bilayer. This is lipid bilayer. This protein is rotating by 180 degree. So when it is rotating, anything is facing upward. After some time it is facing downward. How it is possible? First it is going to this side, slowly, then going to this side, then going to this side, then going to this side, then coming here. So anything moving like this, moving from here to here, it is called which one? Lateral movement. So these lateral movement of the protein occur due to fluid nature of the lipid bilayer. You got the idea? Understand the idea? Clarity came in the mind? What is common about mesosome and plasmid? Both are present in prokaryotic cell. Mesosome is what? Enfoldings of the plasma membrane. Mesosome is what basically? Enfolding of plasma membrane. Plasmid is extracircular DNA. So it is a DNA. It is what basically? Plasma membrane. It is mesosome. So what common is there only in that question? Both are present in which cell? Prokaryotic cell. Now example of blue-green algae, some student did mistake. Example of BGA. In short, blue-green algae is called BGA. Full name I am also writing. Blue-green, example of prokaryotic cell I am writing. I am writing what? Example of prokaryotic cell. Example of prokaryotic cell. See, I am writing many examples. Prokaryotic cell. First I am writing Archaebacteria. Second example I am writing Eubacteria. Third example I am writing Cyanobacteria. Other name of Cyanobacteria is called blue-green algae. Blue-green algae. Another example I am writing Mycoplasma. Got the idea? So these are some of the example of which one? Prokaryotic cells. Many more are there. If you want to know more, you can write Chlamydia, the Rickettsia, Spirochete. These all are example of which cell? Prokaryotic cell. What the idea? Now you are rich in example of which one? Prokaryotic cell. Archaebacteria, Eubacteria, Cyanobacteria, that is also called blue-green algae, Mycoplasma, Chlamydia, Rickettsia, Spirochetes, all are prokaryotic cell. Got the idea? Single membrane bounded, unit membrane we are saying for delimiting membrane, thin membrane, plasma membrane who are covering any organelle. So I told the example of double membrane structure in cell. I am writing once again, double membrane bounded a structure, or you can say double unit membrane. Double unit membrane. You got the idea? Example I am writing. In cell, three structure are there who are double membrane. One structure is nucleus. Another structure is plastid. Another example is mitochondria. These are surrounded by double membrane. Got the idea? Now I am writing example of which one? Single membrane bounded. I told it when I am telling you the flow chart. If you can recall, you can check on YouTube also. It is there. I wrote already in a flow chart format on the board. Got the idea? YouTube not available? Very good. Keep it up. Right. So single membrane bounded organelle is which one? Endoplasmic reticulum, Golgi complex or Golgi body, you can say anything, lysosome, vacuole. Got the idea? These are single membrane bounded structure. Now I am telling non-membrane bounded structure. None means no membrane, non-membrane, non-membranous, non-membranous structure. Centrosome, Other example is ribosome. Got the idea? You got a picture who is double membrane bounded, who is single membrane bounded, who is no membrane bounded cell organelle. Picture came in your mind. Now you will make a mistake. No. So your student are writing chloroplast. Chloroplast I wrote in which one? Chloroplast is a type of plastid. Actually, I will teach you later. Plastid is of three types. Chloroplast, I will teach later. Chloroplast, chromoplast, and leucoplast. Got the idea? These are the type of plastid only. Chloroplast is a type example of plastid, type of plastid. Clear? Done. So cell wall quasi fluid nature. One student asking, कि sir, you are telling plant cell, here it is cell wall, it is rigid, and you are saying, sir, plasma membrane is quasi fluid. This is quasi fluid. This is plasma membrane. This is quasi fluid. It have a fluid nature. Got the idea? It have a fluid nature. Cell wall is what? Thick and rigid. It is solid. It is solid. Who is fluid nature? Cell wall is fluid nature. Wall is what basically? Sorry, solid nature. Wall is solid nature. Plasma membrane is fluid nature. You got the idea? Mesosome itself a membrane. One student asking, mesosome is membranous or non-membranous? It is mesosome. Mesosome is enfolding of plasma membrane. Enfolding of plasma membrane. Did you got the idea? Mesosome is enfolding of plasma membrane. Wall, cell wall is non-living. Plasma membrane is living. I told in beginning when I was teaching cell wall. Non-living. You can recall that lecture. I wrote it is a non-living boundary, and plasma membrane is a living membrane. Got the idea? Is it clear? Any other question? Any other question? Prokaryote also contains cell wall, na dear. You was absent in prokaryotic cell. I have told that prokaryotic cell have three kind of layer. This is a prokaryotic cell. Three kind of boundaries there. Trilamellar structure present in prokaryotic cell. I am telling you the boundary of prokaryotic cell. Third boundary is this. These are prokaryotic cell boundary. Got the idea? So this is outermost boundary of the prokaryotic cell. What we are saying? Glycocalyx. Outermost boundary of the prokaryotic cell. Cell wall is second boundary from outside. And third is cell membrane. Cell membrane, third layer. Enfolding of cell membrane making two type of a structure. When it is trapping pigment, it is called chromatophore. Everything I told, what I asked in paper, chromatophore, and here it is what? Another enfolding mesosome. So this is a prokaryotic cell wall, bacterial cell wall. Bacterial cell. So you see in bacterial cell how many covering is there? Three covering are there. Three boundary is there. Boundary number 1, boundary number 2, boundary number 3. Second boundary is called cell wall. Got the idea? Picture came in the mind? No need to take a screenshot. In last I will share the screen. Got the idea? So you can take a link from, you will got a link through that share. You just scan it, you will got the entire board on your mobile. Don't worry. But I always say prefer to write. I always say what? Prefer to write. See, study is a process. If you are methodical, if you are honest, if you are consistent, if you are patiently listening, continuously attending the class, there is not a single reason you will not understand. But what happened many times? Many times people find they are in home now, listen 5 minutes, 10 minutes, go somewhere, then come, then listen, then go, then come. So, see, a study is not a casual process. Simply through your camera you can scan. I will say in the last. Through your camera you can scan. Okay. Today by scanning I will show you. Don't worry. If anybody don't know to scan, I will say you how to scan. Getting the idea? One student asking, sir, can you tell me about plasmid briefly? Plasmid is what basically? Plasmid is a extracircular genetic material, DNA present in prokaryotic cell. It is a double standard structure. It is extra DNA. It is extra genetic material. It is circular structure. Getting the things or not? It may contain some gene who modified the phenotype of the cell, like antibiotic resistance gene. That line written in NCERT. Got the idea? So can we start the today session, or any more question is there? Can we start the today session? So what I am requesting to all of you, see, 2-3 things you do, you will find you are improving a lot. First thing I am requesting all of you, arrange NCERT for you. If it is hard copy possible, you purchase it. If it is not possible, you can download through Google or through NCERT website or through ePathshala I told. Second thing, listen every lecture. If you are sitting in home with your freedom, it doesn't means, okay, if I want to see, I will. If you don't want to see, I will just simply open it and just, okay, time to time I'm going to go a formality. It is not a formality. You have to be consistently listen the every lecture from first minute to last minute. First minute to last minute. Got the idea? Second thing is listen the lecture. Third thing, you just revise what you note down. Note down the things. What we are writing on the board, make a note. So sit in a proper table and chair and note it. Got the idea? If doubt is coming, you just ask. Getting the things or not? If somehow many doubt is there, bigger doubt is there, when you just note it down. When you came in the campus, ask, or when you people are teaching this, now I got this particular area doubts and I just want it to clear. Getting the things or not? So be consistent, be honest, everything will happen great with you. Right? And you are here, that means it is showing your seriousness. That a great thing. Right? Otherwise, many people are in euphoria of what? Okay, board exam given, I will see later. Yeah. Got the idea? These are the most, I am going to tell you, everyone who are here right now, these are going to become the most toughest time of your life. Most toughest time of your life. Isn't it? Because you are going to crack one of the biggest exam of the country. 10th to 11th class itself, some quantum jump occur. Syllabus difficulty level increase. So you have to fight with that also. Other things, national exam, you have to involve in a big competition as well. What the idea? You have to beat around 24 lakh students. So, right. So that is a challenging task. But any student who are honestly working, definitely they are just going to, and see, now career is a big thing in the life. and to become medical professional, definitely one of the very, very, very noble career, great career. So of course, some inertia is there, but you have to work on that, you have to fight with that, you have to come out. Isn't it? NEET exam is a tough exam in India. It is not a tough exam, but it is highly competitive exam. It is highly competitive exam because you have to fight with 25 lakhs people. Got the idea? It is not tough. If you know your NCERT, if you are listening, you are understanding, nothing a reason you will not become a doctor. Got the idea? Sir, ribosome are present only in prokaryotic soil. No, dear, it is present in both prokaryotic and eukaryotic cell. And many of our students know now in prokaryotic cell, which one is there? 70S type. I told also in eukaryote 80S type. So let's start the today agenda. So I am starting with which one? Endoplasmic reticulum, ER. We are saying what? Endoplasmic reticulum. Right? So what is endoplasmic reticulum? So it is a network or reticulum of tiny tubular structure present inside of cell. So what is endoplasmic reticulum? It is a network of tiny tubular structure present inside of the cell. Got the idea? So I am drawing the diagram of it, so then you will understand it more better. Give your attention. This is a cell. I am just drawing an animal cell, and here it is nucleus. I am not drawing the detailed structure of the nucleus. I just symbolically making a nucleus. So it is plasma membrane, and this one is nucleus. Now I wrote about endoplasmic reticulum. What here? Just give your attention. I wrote here कि it is a network or reticulum of tiny tubular structure present inside of the cell. This is inside of the cell. So inside of the cell, see how the network is formed by the ER. So look here. See. So here you just see. This is the green color structure I just made. What is this green color structure? It is looking like a network or reticulum or not? It is looking like a network or reticulum, na? Yes. So this is a network or reticulum of tiny tubular structure present inside the cell. So this structure is called ER. This structure is called ER. Now you just see this network of tiny tubular structure dividing the cell, internal component of the cell, into two parts. I am just telling one part is this, what I symbolize with cross, and another part I am just saying you, I am just putting a tick mark. Are you getting the things? I am making it somehow bigger. Are you able to visualize it or not? So it is dividing the internal component or compartment of the cell into two parts. One component is present inside of lumen of here. It is lumen of here. This part is called what? Lumen of here. Lumen of endoplasmic reticulum. So the part of the cell which are present inside the lumen of cytoplasm, lumen of endoplasmic reticulum is called luminal. Luminal. Got the idea? So content of the cell which is present inside, when it is present inside the lumen, it is called luminal. And who is not present inside the lumen, it is called extraluminal. And these extraluminal part is called cytoplasm. Got the idea? So cytoplasmic content inside the lumen is luminal, outside the lumen is called extraluminal. You got the idea about it? You got the idea? You able to differentiate between luminal and extraluminal? Got the idea? So inside the entire cell, you will find such kind of network like structure is there. I just shown only one side. This is present all around the cell, such kind of a structure. Understanding the idea? So what benefit occur with this particular arrangement? So these ER, can I say, increase the surface area of cytoplasm? Because I told earlier, any enfolding increase surface area. It increases the surface area of the cytoplasm. Are you getting the idea? Is it clear in the mind? Because it is an enfolded structure, or any kind of enfolding. I told in beginning class, if you can recall, any kind of enfolding increase the surface area. Any enfolding is there anywhere, it means what? Surface area is going to increase, more surface area is there. So I hope till now you understand what is endoplasmic reticulum. Endoplasmic reticulum is a network or reticulum of tiny tubular structure. Getting the things or not? Of course, fluid is present inside this also and outside this also. Got the idea? Is it clear in the mind? Is it all right? So it is dividing the internal part of the cell into two parts, luminal and extraluminal part, and it is increasing the surface area of the cytoplasm. Now I am going to move ahead. Now if we talk about endoplasmic reticulum, it is of two type. It is of two type. I am not dealing much details of the things. Getting the things or not? Because it is not nowadays not going to ask. Otherwise I can say about the type of endoplasmic reticulum, blah blah. But so I am just going to say based on somehow NCERT. So endoplasmic reticulum is two type. One is which one? RER, rough endoplasmic reticulum. Another is which one? SER, smooth endoplasmic reticulum. So what is the basic difference between RER and SER? What is the basic difference between RER and SER? I am just telling you. So RER, if you talk about, present toward nucleus in the cell. If you talk about ACR, it is present toward present toward plasma membrane. So endoplasmic reticulum who are facing the nucleus is called RER, and who are facing toward the plasma membrane is called ACR. Another differences I am saying between the RER and ACR is R means rough. R means rough endoplasmic reticulum and S means smooth endoplasmic reticulum. When you just see a rough endoplasmic reticulum, I am just showing you, when you see it through the electron microscope, you will find this is the surface of membrane of ER. You will find some granular depositions are present on the surface of ER. And due to that granular deposition, it is looking smooth or rough. It is looking rough. What the idea? So this is RER, rough endoplasmic reticulum. Why it is appearing rough? Because ribosome is present over it. Ribosome present on the surface of ER, where ribosome present on surface of ER, it is called RER. And which type of ribosome is present? ATS type of ribosome present. ATS type of ribosome is present. So on the surface of when on the surface of ER ribosome is present, that particular surface is called rough endoplasmic reticulum. See na, it is looking like granules. It is looking like granules and making the surface as rough. So this is about the rough endoplasmic reticulum. Ribosome present on the surface of endoplasmic reticulum. Then such endoplasmic reticulum is called RER. Here I am telling about a smooth endoplasmic reticulum. No ribosome is present. No ribosome is present on ER surface from ACR surface. Its surface looks smooth. This is ACR, smooth endoplasmic reticulum. Are you getting the idea? I am just showing a membrane. It is a single membrane structure, so I am showing simply by which one? Membrane. Got the idea? In this diagram, I am showing RER or ACR. But before that, let me write some more difference between RER and ACR. You got the one idea? RER present toward nucleus, R for rough, S for smooth. Why it is looking rough? Because ribosome is present on its surface. And which type of ribosome? ATS type of ribosome. Got the idea? Here ribosome is not present on its surface. Getting the idea? Now I am just writing some more difference. So it is present, it is means I am telling for RER, it is present abundantly means in large amount in cell associated with protein synthesis. So all the cell who are actively involved in protein synthesis or enzyme synthesis, because most of the enzyme are made up of which one? Protein. So all the cell who are actively involved in protein synthesis or enzyme synthesis, got the idea? Is you will find in that particular cell who is present in large amount. Rough endoplasmic reticulum is present in large amount. Protein synthesis. Protein synthesis, you know, na? I will tell about later on what is protein. So cell involved in protein synthesis, enzyme synthesis, you can say like. So they have large amount of which one? Rough endoplasmic reticulum. Why? Because protein synthesis is occur by which cell organelle? Anybody can answer? Which cell organelle is performing protein synthesis? Which cell organelle is present called is responsible for protein synthesis? Ribosome. ER do not do protein synthesis. Don't confuse yourself. Many times student do mistake here while saying answer. When I ask a question, cell organelle associated with proteins, cell organelle perform protein synthesis, and student give the answer ER. ER not do protein synthesis. Then who do the protein synthesis? Ribosome present on ER perform protein synthesis. Because ribosome, here it is, my hand is which one? ER surface. And this one is assumed ribosome. It is present here. My hand is not doing protein synthesis. This pen marker is doing protein synthesis. This is what basically ribosome, and this is which one? ER. So protein synthesis occur by ribosome present on ER. Are you getting the idea or not? Is it clear? Done? One student asking what is S? I will tell you what is S when I will teach ribosome. Is it clear? So this is involved, this is present in large amount in the cell who are involved in the protein synthesis. If we talk about ACR present in cell involved in lipid synthesis and steroidal hormone synthesis. Present abundantly in cell. I am writing this word also. Present abundantly in cell associated with lipid synthesis and steroidal hormone synthesis. Are you getting the idea or not? So steroid is a kind of which one? Lipid. So the hormone who are steroidal in nature, that is also synthesized by which one? So if you talk about function, it is a very important line going to ask in exam, function of which one? SER. SER function. One function you can write lipid synthesis. Lipid synthesis. Other function you can write steroidal hormone synthesis. Hormone synthesis. Even phospholipid form in which one? Phospholipid synthesis. Phospholipid synthesis. Phospholipid synthesis also performed by which one? Smooth endoplasmic reticulum. Also I am writing one more function of a smooth endoplasmic reticulum. It is written in your 9th book. Detoxification of drug. Probably it is in your 9th NCERT. Is there or not? Detoxification of drug. What the idea? So these are the some of the functions of which one? Smooth endoplasmic reticulum. Are you getting the idea? One function I wrote which one? Lipid synthesis, steroidal hormone synthesis, phospholipid synthesis, detoxification of drug, pollutant, allergent, etc. These are going to detoxified by which one? Smooth endoplasmic reticulum. Understand the idea? Is it clear in the mind? Okay. So now I am writing overall function of ER, a combined function of ER. You want this slide once? I am telling about function of ER, and here I will write a combined function. Endoplasmic reticulum. First function I am writing, provide support to cell. But these words not written in NCERT, use it wisely. How it provides the support? Because it is a network like a structure. Got the idea? Another second things I am writing is the function is intracellular transport. Transport within the cell. I am just telling you through the diagram. Then you will understand it. According to NCERT over, I am just now going to say something more than NCERT. Now something from nucleus, they can transport it from one part of cell to another part of the cell and even out of the cell. Getting the things or not? With the help of which one? These ER, because they are interconnected. Getting the idea or not? Understanding the idea? And why I am telling it is helping in support as well. One side attached with which one? Nuclear membrane, another side attached with which one? Plasma membrane. So these network like a structure also have some role in support of the cell. But these things not written in NCERT. Now I am just writing, telling you another things. Some more functions I am writing. I am writing associated with. Associated, I am writing a wise word. I am not writing its function is protein synthesis. Associated with protein synthesis. Because ribosome attached with it. Protein synthesis. And even I am making it more clear due to attachment of ribosome on its surface. On its surface. Another function I am writing, lipid and steroidal hormone synthesis. And why I am teaching more than NCERT here? There is a reason. If you go for last 8-year question, in this year, sometime NEET paper cross the NCERT boundary. But good thing is what? From last year they are not asking anything out. Last 4 years they are not asking anything out of NCERT. But for mental satisfaction, okay, earlier it came and I will handle even P by Q. Previous year question, I am telling you, but nowadays that much function not going to ask. What going to written in NCERT, going to ask in exam. So no need to worry much. So lipid and steroidal hormone synthesis. Another function I am writing, phospholipid synthesis. Phospholipid synthesis. Got the idea? Another functions I am writing, and especially I wrote phospholipid earlier. I wrote this is the job done by which one? ACR. This is the done by which one? ACR. What the idea? These are associated with which one? RER. Understanding the idea or not? If we talk about another function, I am writing once again, detoxification of drug. And this is also done by which one? ACR. You can write another function, formation of Golgi body. Golgi body is also formed by which one? Formation of Golgi body. This is also done by ER. Getting the things? Golgi body formation occur also by which one? ER. Another function you can write, if you wish, folding of protein. Folding of protein. What is folding of protein? You will understand when you understand the protein structure in biomolecule. Meanwhile, you simply write folding of protein is also occur in which one? Endoplasmic reticulum. Folding of protein is also a function of which one? Endoplasmic reticulum. And with a star mark I am writing glycosylation. Glycosylation. Glycosylation mainly occur in Golgi body but also occur in ER. Once a question came in NEET कि glycosylation occur in which cell organelle, and it is not given which one as a option, Golgi body, it is given ER as a option. So glycosylation is what basically formation of glycoprotein and glycolipid. Glycolipid. Got the idea? Now anybody asking ki formation of Golgi body done by which ER? Usually they not ask. And if anybody asks you to differentiate between RER and ACR, you can say about which one? ACR. Because neither on Golgi body ribosome is there nor on smooth endoplasmic ribosome is there. Nobody will ask, but anybody if it is asking, you can answer like this. Got the idea? And folding of protein synthesis occur in which one? Rough endoplasmic reticulum. So it is undergoing in folding in there only. Getting the idea? Understanding the idea? So you got the somehow the detailed function of ER. Right. So you studied about sarcoplasmic reticulum in muscles. So sarcoplasmic reticulum is a modified ER. In zoology you will study about this. Is a modified ER. Desmotubule, I told it in plasmodesmata, right now? Desmotubule is also a modification of ER. So I told about ER much more than NCERT. Now there is no limit of access. You can teach entire class or entire 2 days. So definitely we are stopping here on the ER. Otherwise access don't have any limit. Access is limitless. Got the idea? What is glycoprotein and glycolipid? Glycoprotein is made up of carbohydrate plus protein. When carbohydrate join with protein, it is called glycoprotein. And glycolipid, it is carbohydrate plus lipid. Carbohydrate plus lipid is called glycolipid. You got the idea? Glycoprotein and glycolipid. Okay. So understand about the function of endoplasmic reticulum. Is it clear? So once again I am giving you a revision of ER because it is an important cell organelle. So just have a look. Because there is no time, we have to start the new cell organelle. So I am just giving you a brief revision. Listen it carefully. I just request you one thing from my every student. Be in the class for the last minute. I am also hungry. Right? You are not only hungry. But you have to show more focus because you are going to become a doctor. It is your life, it is your exam. We are only the catalyst who will support you, who have to work. Got the idea? Both any enfolding, not ER. I told any enfolding increase the surface area. Meaning of detoxification. Detox means any poisonous things is there going to minimize it. That is detoxification. Impact of reducing the impact of toxic material is called detoxification. So endoplasmic reticulum, I told it is a network or reticulum of tiny tubular structure dividing the component inside the cell into two parts, intraluminal or extraluminal. Inside the lumen, outside the lumen. So normally extraluminal part we are telling cytoplasm. One side here attached with nuclear membrane, another side attached with plasma membrane. So ER who are present toward nucleus have ribosome on its surface. What the idea? Present toward nucleus have ribosome on its surface. It is called RER, rough endoplasmic reticulum. Toward the plasma membrane, ribosome is not there. It is called smooth endoplasmic reticulum. So here I wrote some difference between RER and SER. RER present toward nucleus, SER present toward plasma membrane. Ribosome is present, not present. It is associated with cell, actively involved in protein synthesis. It is present in large amount in the cell associated with lipid synthesis. Function, especially I wrote here for ACR. What I wrote the function for ACR? Lipid synthesis, steroidal hormone synthesis, phospholipid synthesis, detoxification of drug, allergen, pollutant, etc. Got the idea? Then I wrote a combined functions here of the endoplasmic reticulum. 9-10 things I wrote here. What the things I wrote in the ER? Provide support to the cell. The cell organelle who provides support is which one? ER. Intracellular transport associated with protein synthesis due to attachment of ribosome. It not do protein synthesis, ribosome do it. It do lipid and steroidal hormone synthesis. So directly I wrote lipid and steroidal hormone synthesis associated with which one? ACR, phospholipid synthesis, ACR, detoxification of drug, ACR, formation of Golgi body, especially I wrote ACR. No one will ask, but if anybody asking to differentiate either RER or ACR, go with it. Folding of protein, glycosylation means formation of glycoprotein and glycolipid. It is mainly done by Golgi body, but it also perform. Got the idea? Right? And I told sarcoplasmic reticulum is a modified ER, and later on I wrote descemet tubule also a modified ER. Now I am sharing QR code. So now I told you now how to scan it also I am telling you. So just simply open your camera and scan the QR code like this. Wave address, something like this came in your mobile. You click it. Entire notes came in my mobile. When gift class will end, I also don't have the idea about the exact date. I will discuss with the management and I will tell you. Got the idea? So I don't know either scan in Google or not. You can scan through your camera. I just scan it. You can check. It is there in my mobile now. What I wrote in my mobile now. Everything came. Simply through a camera you can scan. I scan the QR code by opening my camera only. I just open camera for clicking the photo and putting the camera here. Simply QR code link came. Okay? Understood? Clarity came in the mind? Okay, thank you very much. Have a great day for all of you. So offline it will open, probably in the last week of May it will open. It may be from 28th of May, I'm not very sure about the exact date, but it may like in the last week of May it will open. Got the idea? Sir, are the lipid synthesized by RER? Lipid not synthesized by RER. Lipid synthesized by ACR. Protein synthesis associated which one? RER. Got the idea? No problem. Any NCRT you can bring with you. Old NCRT, new NCRT, old NCRT have some more information. You can go for it. No problem. Got the idea? Because nothing is extra given in new, something deleted. Okay. Right. So I will ask them to make an exclusive group for you. Don't worry. Then you can contact. Okay. Thank you. Thank you very much. Bye. Have a great day to all of you. Bye bye. Thank you very much for your patience, for listening. Keep it up.